Hello, Naomi. Welcome to our video lecture for Chapter 10, Blood. So for this chapter, we're going to take the approach of hitting the highlights and talk about the main topics and main characteristics of blood in this chapter. We're going to keep this to one video because we're only going to hit the highlights. So here we go, Chapter 10, Blood. So as we did in Chapter 3, when we discussed the four different types of body tissues, we discussed that blood is a connective tissue. Blood is the only tissue in the body that is a fluid. The two main components of blood are called the cellular portion, living cells, which are called the formed elements, and the non-living matrix, which is sort of like the material that the cells are embedded in. In this case, with blood, it's called plasma. So the fluid part of blood is called plasma. The cellular part of blood are called formed elements. More about that right now. So this picture right here, I'm going to skip ahead just to show this picture bigger, then I'll go back to the notes. So if you were to take a blood sample from an individual and centrifuge it or spin it down in a centrifuge really fast, you get what looks like this. The heavier layer, the denser layer is the red layer. You actually can get a little red pellet of red blood cells. Erythrocytes are red blood cells. That's the red part of the blood. Then you get this thin layer called the buffy coat. This contains leukocytes or white blood cells and platelets, which we'll discuss in a minute. Combined, those are called formed elements. So the cellular part of blood are red blood cells, white blood cells, and platelets. The top layer is plasma, which again, you can see the percentages, 45%, less than 1%, 55%. Plasma is the fluid part of blood. It contains ions, electrolytes, water. It has sort of this lightish, almost sort of yellowish, clearish, yellowish color. That's called blood plasma. So going back to the page where we had the note. So if you centrifuge blood, erythrocytes, which are red blood cells, sink to the bottom. Hematocrit is what you call that bottom part of blood when you centrifuge it, the red part. The buffy coat, again, contains leukocytes, which are white blood cells, and platelets, less than 1% of your blood are those two. And the top layer, the plasma layer, is about 55% of your blood, is largely water and electrolytes. More about each layer in just a minute. So this is a diagram from the book. So, you don't need to have these numbers memorized. Red blood cells, this is the number per cubic millimeter of blood. So picture a millimeter, a tenth of a centimeter as a cube. It's very, very small. Between 4 and 6 million red blood cells per cubic millimeter. Red blood cells, we'll see in a minute, transport oxygen and carbon dioxide through your body. Leukocytes are white blood cells, much lower numbers, between about 5 and 10,000. This is for defense and immunity, your immune system. The different types, basophils, neutrophils, eosinophils, monocytes, and lymphocytes. More about those in a minute, but those are all white blood cells. And platelets, you can look at the numbers, very, very high. Platelets are cell fragments, and they're involved in the clotting of blood when you have a cut or an injury. So physical characteristics of blood. So back when we did, I forget the chapter, when I harped on the fact that blood is never blue. I think I showed this picture. Maybe I didn't, maybe I didn't. So, there's this notion, someone in your past has taught you that blood is blue. Blood is never blue. I think I've already harped on this. In textbook diagrams, sometimes they draw venous blood that's in veins is blue because they draw the veins as being blue. But blood is never blue. So blood varies from a bright scarlet red to a dull, sort of approaching dark purple red. Arterial blood, and when we do chapter 11, we'll talk about obviously the cardiovascular system in more detail, where our arterial blood is the blood that's in arteries, which is very oxygenated. Venous blood is the blood that's in your veins, which has lost its oxygen to your tissues. And you can see the range of colors from bright scarlet to a dull red. The pH of blood is very, very, very slightly basic, 7.34 to 7.45. If your blood pH gets out of balance, you go into shock. It's a bad thing. You read in the chapter 1 of Serengeti Rules about Dr. Cannon, the guy that figured out the whole deal with bicarbonate in World War I. And if you give someone bicarbonate, you help lower the incidence of shock because your blood's becoming too acidic. when you go into shock, you see the blood pH is very slightly acidic. I'm sorry, very slightly basic. And if you vary that range too much, you have a very, very bad day. Your blood tip is slightly higher than average body temperature of, in this case, about 100.4 degrees Fahrenheit. You contain, you can read this, and a guy between 5 and 6 liters, or about 6 quarts. I'm not sure what it is for a girl. I need to Google what it is for a girl. I'll do that and add it to the video right here once I finish recording it. By weight, blood is about 8% of your body weight. The blood plasma, remember that was the sort of yellowish layer at the top, about 90% water. You can see the things that are in blood plasma. So, nutrients that your blood carries, salts, which are called electrolytes, respiratory gases, oxygen, carbon dioxide, hormones, your blood carries hormones from chapter 9, plasma proteins, waste products are all carried in the plasma part of your blood. The formed element portion, the cellular portion, has 3 main parts: red blood cells, white blood cells, and platelets. Erythrocytes are red blood cells, leukocytes are white blood cells, and platelets, again, are cell fragments involved in clotting. So more on each of the types. So red blood cells, their main job is to carry oxygen. If you look at pictures, this is a nice 3D picture. When we looked at blood under the microscope, it looked more like this. You saw more of a 2-dimensional image. So they're biconcave discs. Concave means to bow inward. So they're discs where both sides bow inward. They're essentially bags of hemoglobin. Hemoglobin is a respiratory pigment. It's actually red. It's based upon the element iron. And hemoglobin carries oxygen through your body. Without hemoglobin, you don't really, it. How does oxygen go from your lungs to your blood to your tissues? Hemoglobin is what's carrying it. Erythrocytes are anucleate, as in they don't have a nucleus. They're not very long lived. They only live about 100 or so days. They have very few organelles. Once you produce them, they carry hemoglobin, oxygen, for about 100 days, and they die. There are about 5 million red blood cells per cubic millimeter of blood, which that's a lot, right? So when we looked at blood under the microscope, normal blood, it looks sort of like this. The bluish purple cells are different kinds of white blood cells, which we'll see in a minute. The little fragments of things are platelets. And then everything else that you see are red blood cells. And you can kind of see that they look kind of like donuts because they're the biconcave disc. So hemoglobin, like I said, it's an iron-containing protein. It's a respiratory pigment. So hemoglobin, the second point says binds strongly but reversibly to oxygen. Hemoglobin binds oxygen, which that's its job because it's going to take oxygen to your body, but it also releases the oxygen. So think of in your lungs, the blood capillaries in your lungs, which we're getting a little bit ahead of ourselves, so the respiratory system in this case, but in your lungs, there's lots of oxygen, right? So hemoglobin binds oxygen pretty tightly in your lungs, but as your blood goes through your body, once your blood, say, gets to your pinky toe, there's less oxygen because your body is using it. So in your pinky toe, hemoglobin releases oxygen to your tissues, okay? Every hemoglobin molecule has 4 oxygen binding sites. So a hemoglobin molecule can carry 1 oxygen, or 2, or 3, or 4, or none. So in your lungs, it's probably carrying 4. It's uploading oxygen. In your pinky toe, in your tissues, it's probably carrying, the 3 to 2 to 1 to maybe 0 because it's unloading the oxygen. Each red blood cell has 250 million hemoglobin molecules. Don't worry about memorizing that number, but they have a lot. And normal blood contains about 12 to 18 grams of hemoglobin per 100 mils of blood. If your hemoglobin levels get too high or too low, that's your red blood cell, your red count. Obviously, that's a problem. They can do a finger prick to see what your red count is. Actually, well, what's the problem? Here's a couple of problems. So homeostatic imbalances. So anemia would be, if for whatever reason, if your red count is low and the ability of your blood to carry oxygen goes down. If you are anemic, you're sluggish, you're fatigued. You could be anemic because you are bleeding somewhere. Sometimes when a girl is menstruating, she might become anemic. Medications could cause it, cancer could cause it. That's anemia. Low iron in your diet can cause it. Sickle cell anemia or sickle cell disease is caused, you probably learned about this in ninth grade, abnormally shaped hemoglobin. So here's a nice biconcave disc of hemoglobin. Sickle cell disease is, it's one base of your DNA, one A that should be a T, or I think it's an A that becomes a T. I forget the actual letter, but it's one point mutation in your hemoglobin gene where a glutamine, amino acid in position 6, is a valine. That's it's one tiny change, one letter of the alphabet of your DNA is wrong, but it has a huge effect. It causes the red blood cells to form this sickle shape, which means they don't carry oxygen as efficiently. And it's a very, very, very painful disease, especially around joints. It disrupts your blood flow. And around joints, you can have chronic pain. People that have sickle cell disease might be in the ER in the hospital a lot, obviously no fault of their own. The only way to really treat it or to cure it would be a bone marrow transplant would do it. Now, in the last couple of years, there's been advances in gene therapy where you actually use a virus to go in and fix an individual's genes, because we know what the correct gene looks like. Most of you have the correct gene, and actually fix it genetically. And that's been very successful in virtually curing people's sickle cell disease by genetically altering their blood cells so they have the right form of hemoglobin. So leukocytes are second type of formed element. These are white blood cells. So this is your immune system, right? White blood cells are complete cells. They're not like red blood cells that are lacking a nucleus or organelles. These have a nucleus, they have organelles. They're very vibrant cells. They can do lots of things. Diapedesis, this word here, white blood cells can do something that's really kind of wicked. They can actually like leave blood vessels and go into your tissues, into the interstitial spaces between your cells. They move by amoeboid movement. Picture like an amoeba that you might have learned about in middle school or ninth grade. Amoebas do have the pseudopods that do it's called cytoplasmic streaming, how the pseudopods kind of like, I'm not describing this well, am I? They kind of like ooze around. They can move by amoeboid movement. They can respond to chemicals released by damaged tissues, chemicals released by cells that are infected with a virus or a bacteria. They can kill cells, destroy cells, cause an inflammatory response. More about the immune system, obviously, later. It's a whole chapter all by itself. But they're white blood cells. They're between 5 and 10,000 or so white blood cells per cubic millimeter of blood. Reasons why your white count would be abnormal. So leukocytosis would be if your white count is too high. So, the number on the previous slide, the 5 to 10,000 or so, that's a normal person. That's like, you're not sick. That's you're having a fine day. If your white count's too high, well, that's a problem. I mean, something's going on there, right? It could be that you have an infection. Maybe you're sick. Maybe you have the flu, or you just have a cold or strep throat or whatever, and your white count's high because you're fighting off a disease. That's totally reasonable, right? If your white count's too low, leukopenia would be probably because some kind of medication's doing it, like chemotherapy drugs. Chemotherapy kills cells that divide rapidly, those are cancer cells, right? Well, white blood cells can divide rapidly too. So if you're on chemotherapy drugs or corticosteroids, remember in chapter 9, cortisol, the hormone that kind of like calms the stress reaction, like Portate cream if you have a rash, those kind of suppress the immune system. I mean, chemotherapy drugs are killing cells. That might result in a low white count. Leukemia would be if your white count's too high because you have some kind of cancer in your bone marrow, or leukemia is cancer of leukocytes, right? Where the cells are cancerous, so they're dividing way too rapidly, so you have way too many of them. So the different types of white blood cells, we're going to go through these in a little activity in class in more detail. I don't want you to stress over the 5 types. You do sort of need to know them, but right now let's just name them and move on. So from most to least abundant, the cells are called neutrophils, lymphocytes, monocytes, eosinophils, and basophils, okay? No one's ever asked me as a student, name them from highest to lowest, but the never let monkeys eat bananas, which is being very, very mean to monkeys. I think we should let monkeys eat bananas. But if someone said, what's the most common one? It's neutrophils. What's the least common one? Is basophils. What each of these do, we will talk about in an activity in class. But yeah, never let monkeys eat bananas, or the 5 types of white blood cells. More about that later. This just shows, where they come from. We'll talk about this more in class. Right now, don't worry about this slide. So formed elements are the third type of formed elements are platelets. Megakaryocytes are what you call where they come from. These are multinucleate cells that you basically just kind of rupture. They are involved in the clotting process. If you have a cut or a wound and your blood needs to clot, you can see platelet counts. Don't worry about the numbers, but they range from 150,000 to 400,000 per cubic millimeter of blood. 300,000 is an average for a normal person. Of course, if your platelet level is too low, that could be hemophilia, where your blood doesn't clot, where your blood's too thin. You could be on blood thinners like aspirin. If your platelet count was too low, you wouldn't be on blood thinners, right? But if you are on blood thinners, the count might be low. If it's too high, that's a problem, because then you might get blood clots that are traveling where they shouldn't be. Like a blood clot really anywhere that shouldn't be there is bad. But if you have a wound, you want the platelet count to increase so that you clot and heal a wound. Formation of erythrocytes, just a word. So these can't grow on their own. They come from bone marrow. They last about 100 to 120 days, so only a couple months, or a few months, I should say. So when red blood cells die, they die. They only live 100 or 120 days. Either your spleen or your liver filters them out. The main thing that your spleen does is filters out red dead blood cells, and you can live without a spleen. If you're having some kind of blood issues, the spleen might become enlarged and could rupture, which obviously would be bad. So people can have their spleens removed and live okay, because your liver can do this too. They replace, again, in your red bone marrow by cells of your bone marrow, which again make red blood cells. So you've done this before, so let's just quickly talk about human blood groups. I'm going to actually skip this and go to a picture and then go back to the slide. So there's many different types of blood typing systems. The previous slide says there's over 30 of them. The 2 main ones that we discussed are the ABO group and the Rh factor, which we're going to go through those briefly. So the word antigen. So an antigen is just something in your body that can elicit an immune response. It's an antigen. More about that when we do the immune system. So look at these 4 types of red blood cells. So the A antigen in this picture is shown as circles. The B antigen are these little squares or diamonds. If you're AB, you have both of them. If you're O, you have none of them. So your red blood cells have these surface features on the plasma membrane. They're just proteins or glycoproteins. They're just like markers that tag the blood with whatever antigen that you have. In this case, if you're infected by a bacteria, your immune system recognizes foreign antigens on that bacteria and destroys them. In this case, you don't want to target these because these are your own cells. This is the way of your body recognizing what's called self from non-self. And your blood is self. Those are your cells. Your immune system should not react to it, and they don't. So the A antigen, you only have A. The B, you have B. AB, you have both. They're different, but you have both. O, you don't have any. This one shows little spikes. Honestly, there shouldn't be any antigens on it at all. Now, antibodies are things that your body makes to fight off foreign antigens. So if you're type A blood, you make what are called anti-B antibodies because you don't have B blood. So to you, B is foreign. If you have type B blood, you make anti-A because A is foreign. If you're AB, you don't make any because nothing is foreign. If you're O, you make both anti-A and anti-B. Again, the antigens in your blood, this row is really the same as this row. A has A, B has B, AB has both, O has none. Okay, so let me go back to the previous slide for a second. So you have these genetically determined proteins, it's inherited, called antigens on your red blood cells. Antigens are a way that your body recognizes what should be there and what should not be there. We type the blood using antibodies that will see what responds. If your blood responds to the anti-A antibodies, what's your blood type? Type A. If you respond to anti-A and anti-B, by respond I mean clump, coagulate, then you have type AB blood. If your blood doesn't respond to any of the antibodies, then you have O blood. Again, there's over 30 different common red blood cell antigens. The main one that we talk about is the ABO group and the RH group. Okay, we've already said all this. Move my face real quick. The ABO group, you have A, you have B. You can have one of them, both of them, none of them. If you have none of them, then you have type O blood. This is what we said just a minute ago. So in terms of what you can receive and what you can donate, so you can only receive, let me rephrase that. You can't receive what you don't have. You can't receive what's foreign. If you have blood type AB, you have everything. You have A and B. You can receive A, AB, B, or O. So having AB means you are a universal recipient. You're lucky, right? If you have type O blood, you look at the bottom, if you have O, A, B, and AB are foreign to you. So you can't receive anything except for type O blood. And of course, B can receive A or O. Wait, did I say that right? B can receive B or O. A can receive A or O. All right, so you can memorize this or just think, you can't receive what you don't have. You can't receive what's foreign, or your immune system will attack the blood transfusion and will cause the blood to clot, which obviously is bad. This is just from your book. This is just a review. AB has A and B, makes no antibodies. You can see what you can take. In class, we're going to do a little handout on this in more detail. So for right now, let's just kind of put a pause on this and come back to this later. If you're O, you have none. You make all the antibodies, and you can only receive O, but you can donate to anyone. So what's the deal with the Rh factor? So this is the deal with like A positive or O negative. So the Rh factor was first identified in rhesus monkeys, which is why it's called the Rh factor. This is a different antigen on your blood cells. And basically either you have it or you don't have it. It's actually 8 different antigens, but long story short, you have it or you don't, right? Rh positive means you have it. Rh negative means you don't. And there's a problem if you mix the two bloods together, it's going to clot, because if you are Rh negative, you don't have the Rh factor. If I give you Rh positive blood, guess what? That's foreign, so it's going to clot. So if I'm positive, you can give me negative because that's not foreign to me. But if I'm negative, you can't give me positive because that's foreign. All right, so reviewing ABO and Rh factor together is this little diagram. So this individual has everything. So Rh is a black square, A is a tan, orange, yellow, whatever color that is, triangle, B is a blue triangle. So AB positive has everything, all right? What can AB positive receive? It can receive everything. That's the true universal recipient because nothing is foreign. All right, on the other end, O negative, you have nothing. So what can you receive? You can only receive O negative because everything to you is foreign except for O negative. So looking at the diagrams, like basically you can receive anything that you have. So an AB positive person can receive, list them all, AB positive, AB negative, A positive, B negative. I'm not going to name them all, but you get the idea. Everything that's listed here. So look at A positive. What can A positive not receive? And A positive cannot receive anything with B. So an A positive cannot receive AB positive, AB negative, B positive, B negative. If it has a B, you can't receive it. All right. Let's do another one. AB negative, what can they not receive? They can't receive anything that has a positive. So AB negative can't receive AB positive, A positive, B positive, or O positive. All right. Again, you can only receive what you have. You cannot receive what is foreign. So this comes to a real world application. So Rh dangerous during pregnancy. So this is actually a big deal. So what if a mom and a child have different blood types? Which that happens a lot, right? It's actually would be unusual for the mom, the baby to have the same blood type because the dad obviously contributes some genes to the kid too, right? So the mom's blood and the baby's blood, we'll do this in chapter 16, they don't actually mix. There's no blood literally mixing together. Basically in the placenta, the mom's capillaries and the baby's capillaries kind of intermingle and blood doesn't go between them because that would be a really bad idea because they're probably different blood types. But things can go between them, like glucose can go between them, nutrients can go between them, because obviously the oxygen, because the baby's not breathing, baby gets oxygen from the mother's blood supply. But antibodies occasionally can go between them. So if you have a mom who is Rh negative and is pregnant with a baby that's Rh positive, again, the blood itself doesn't mix, but antibodies between the blood can mix, and the mom could start producing antibodies to the Rh factor, anti-Rh antibodies. Yes, that's how you say that. Usually, the first time that happens isn't a big deal because it takes time for the effect to happen to build up some immunity. So the first time that an Rh negative mother has an Rh positive baby is not usually a problem. But if that happens again, if there's a second pregnancy where the mom is negative, same mom, so negative, and the baby is positive, that could result in a miscarriage. There's a shot that you give mothers called the RhoGAM shot. It's very, very commonly given. I think nowadays they pretty much just give it to everybody, which they would give a mother who is Rh negative after she gave birth to an Rh positive baby. Basically, it's a shot that neutralizes the anti-Rh factor antibodies. My mom is Rh negative. My older brother is Rh positive. So she obviously got the RhoGAM shot. I mean, my brother's in his mid 40s, so 40 some odd years ago. And then I came along. Now I'm Rh negative. So if she hadn't gotten that shot, it wouldn't have been a problem because I'm not Rh positive. But you give the shot as a preventative in case of a second pregnancy, if the mom is Rh negative and the baby is Rh positive. Okay, cool. I already said everything on the slide, right? I mean, if you need to write this down, write it down. The mismatch of an Rh negative mom with an Rh positive baby can cause a problem, not so much with the first pregnancy, but during a second pregnancy, it can happen. The mom's immune system can attack the Rh positive blood of the baby through the placenta. It's called hemolytic disease, but the RhoGAM shot neutralizes that. So how you do blood typing, here's a quick picture. Like this is blood clotting, this is blood not clotting. So you make anti-A and anti-B sera. So basically you can just take blood plasma. If I'm type A, I make anti-B antibodies, right? So take blood plasma for me and use it to make what are called blood sera or anti-A and anti-B sera. Coagulate means to clot. So if it's clotting, like if I have a blood sample and it's clotting with the anti-A sera, then it's blood type A. If it clots with anti-B and anti-A, it's AB. You get the idea. And obviously, you would want to do this before you gave someone blood. If you know the person's blood type, you should all. If you don't know your blood type, go home, ask your parents, what's your blood type? If they don't know, which a lot of parents don't, next time you have your blood taken or a finger prick, make sure that you ask the doctor what your blood type is. You might can log into like your MyChart. It's probably somewhere in your medical records anyway. Your doctor knows what it is. He can look it up. He or she can look it up. But you should know your blood type. But basically what it looks like. So here's, this person has type AB blood because it's clotting with anti-A and anti-B. This person has type B blood. It reacts to anti-B, but not anti-A. This person has type A blood. You see it's clotting with anti-A, not anti-B. This person's type O blood. Nothing is clotting. So you don't have any of the antigens, so you are type O. Okay, so last thing, let's wrap this up. So developmental aspects of blood. So when you're a fetus, your liver and your spleen produce red blood cells. When you're an adult, look at this, when you're a 7-month fetus, once you're approaching being born, your bone marrow takes over. Hematopoiesis, that's a fun word to say 10 times. Hematopoiesis is just the name for blood cell production. And of course, your bone marrow does that. Fetal hemoglobin differs from hemoglobin produced after birth. So the mom has hemoglobin, right? Duh. Well, the baby has hemoglobin too, because the baby, the fetus in the womb, the heart's beating, the blood's pumping. Fetal hemoglobin actually binds, it's a little bit funny, it binds oxygen stronger than the mom's hemoglobin. So the baby literally like rips the hemoglobin or rips the oxygen from the mom's hemoglobin in the placenta and takes it for itself. But that kind of makes sense because the mom can breathe, the mom can get more oxygen, the baby cannot, because it's in the womb. You don't produce fetal hemoglobin unless you're a fetus. So like you have the genes for it your whole life, but once you're born, you stop making fetal hemoglobin because you're not competing with your mom for oxygen. You have normal adult hemoglobin. Physiologic jaundice. So this is when like a baby is born that has kind of a yellowish tinge to it. Jaundice, so you had different biochemical pathways where you break down hemoglobin from blood cells that are dead. If that process is slowed or inhibited, your skin turns yellow, the whites of your eyes turn yellow. Babies can oftentimes be born with jaundice, which is when they're just a little bit slow at metabolizing dead red blood cells and hemoglobin. My daughter, when she was born, babies that are born with jaundice, they put them under, it looks like a tanning bed. Like it's in the nursery. It's a little like heat lamp thingy that they sit in, and the light helps break down the hemoglobin. Again, it looks like they wear little goggles. It looks like they're in a tanning bed, but it's not. It's just a way to break down the excess hemoglobin to get rid of the jaundice. Okay, so that was chapter 10. We got it done in one video. So hope that was helpful, and I will see you guys next time.